pointers, just because they're required. You've already heard those. We've got some objectives, and since I'm the outsider, the geneticist among the endocrinologists, it's best that we all have a common language. So I'm going to try to start with some molecular testing as used in pediatric endocrinology, give you some options for a best genetic testing strategy, not necessarily the only one. Hopefully some tools to be able to interpret variants of uncertain significance in a better way. Some pointers on how to translate genetic results for your families and maybe get you thinking about revising how you do genetic testing. So quick outline, the applications, the role of molecular testing in an endocrinology clinic, what's appropriate testing, where are the guidelines? We always like guidelines. What are the results and the balancing of your expectations and reality? So what's the scope? So what's the scope of genetic disease? Well, many of the endocrine disorders, especially in the pediatric population, are monogenic. So one bad gene or two copies of a bad gene have disease. It is heritable, not necessarily inherited. Heritable, it can be translated to the next generation if your patient survives. They can be dominant, recessive, X-linked. They can even be mitochondrial. That's a whole different topic. Most monogenic disorders are high, which means that there is some component of the phenotype, not necessarily the entire phenotype, due to a variation in the gene. And the penetrance can vary. One good example, common RET sequence change, position 804 in a peptide, valine to methionine, only has 10% penetrance. But most of the other single nucleotide variants in that gene have greater than 90% penetrance, causing disease. Expressivity can vary. So what you see from a single variant can be a spectrum of disease. You can see that from within a family or across families. But in general, monogenic disorders, large effects from a single variant. You can have multiple genes giving you a given phenotype. It's phenocopy. So best example I could come up with quickly was pheochromocytoma paraganglioma disease. There's these 15 genes involved, and we don't have them all because there's still a number of patients that we never get the answer for. We now have the lovely freeze. We've got freeze. So there are also multifactorial genetic endocrine diseases where you often get small effects from multiple genes. This is more commonly thought of in the adult population, but you can have larger contributions from the environment, chemical exposures, lifestyle, epigenetic kinds of things. Your mom was exposed to something, your grandfather was exposed to something, and it causes a phenotype in you. And there's a much larger probability that in the cases of these multi-gene 2 phenotype, that you've got multiple interactions, and it's much more difficult to sort out what any given sequence variants may mean. So how about molecular genetics in pediatric endocrinology? What we've learned has been moved very rapidly from specialty labs to subspecialty care. It's not standard of care yet in the common pediatrician or GP's office. They've gotten faster, they've gotten less expensive. Just a few years ago, a clinical exome sequencing was $26,000. Now it's $2,600. So there's been evolution. We started off with molecular testing being restriction fragment polymorphisms. So it was one polymorphism in one gene, and you got it back a few months later. We moved to Sanger sequencing, where we were doing sequencing of a given gene. Then we moved to multiplexing that, which gets targeted panels or multiple sequence variants within a given gene. Or we do it across many exons at the same time, call it a clinical exome. or you do it on DNA instead of on RNA, and you get genomic sequencing. So one good example of Modi, there's two fairly recent papers, relatively big numbers of individuals, one's 1,100. So, and the other one's almost 4,000 individuals, and they did it reasonably well. They got rid of things that were antibody positive, so they sorted out just like we all would do for those that are at high risk for having Modi. And then they did a 14 gene panel in Qatar. What did they find? They found a few patients that they could identify, a few who had syndromes that probably need other services involved, but they had those ugly 3 VUSs, KLF11 and PDX1. The Swedish study didn't sample everybody. It was more of a convenient sample, so it wasn't set up that everybody got tested. But they found 46 cases of Modi. They only did 3 genes. And they found out of their 3 gene panel, some, but not all of the individuals who truly would have Modi. They didn't disclose the number of VUSs found. So what's the cost effectiveness? We all know that Modi can be misdiagnosed as DM1 or DM2. That's how they get to, it's not quite the right diagnosis. Your genetic testing may be limited by your restricted insurance coverage. Certain insurances just don't believe in DNA. So there's a simulation modeling, typical pediatric population, 10 to 20-year-olds. They did biomarker screens, so they got rid of anybody with an antibody, et cetera, et cetera. What do you have left? And they only did 3 genes. But look at the price of their projected panel, $3,700. Right now, I can walk over to my computer, fire it up, and get that panel, including the cost of counseling the family, and do it for $1,700. So I think their estimates for costs are high on that. They made some reasonable assumptions. 1.2% of the individuals with diabetes having Modi fits what we've heard over the day. And then they did cascade testing predictions for family. What they gained, 0.0042 quality adjusted life years. Quality adjusted life year is a good health statistic for how much better off are you for how much longer. If you get 0.1 quality adjusted life year on a drug, the FDA will approve it. That's a significant change. What do they gain? $191. What would you gain if you actually went out, got the test cheaper, maybe $2,000 per patient over 30 years? Now you're talking almost real money for doing it the right way. Okay. But if you do more sequencing, you of course get more variants. RFLP was easy. It was yes or no. Your patient either had that polymorph or they didn't, and the interpretation was 100%. With Sanger sequencing, you took finding novel variants out of the research lab and put them in front of the clinician. Now you have the problems. Not all variants cause a phenotype, and you might find variants without having a phenotype. Oops. Or you might have variants with more than one phenotype, more than one effect, and they may have other interactions. So a good example again, endocrine example, for RET, it makes a difference whether you have a loss of function mutation or a gain function mutation. Because in one case, you can turf them to your colleagues in GI, case done, and in the other case, you've got to keep them and probably keep the whole family. So it's as simple as who, what, when, where, and how. It's why do you want to obtain a molecular test? Number one reason, you want to diagnose somebody. You want the answer. It's the big 4 questions. What do I have? How do I get it? How are you going to treat it? And who else is at risk? Those are the 4 questions. Those are the only 4 questions in medicine. Okay? So your diagnosis, it may affect management. As we heard earlier today, some management strategies change when you know what sequence variant you're dealing with in which gene. Prognosis, what's the future hold? They may not have disease today, will they have disease tomorrow? When is it needed? If you're diagnosing a proband, you may want it now. By definition, the proband is the one in front of you who's got something most cases. And then who requests this? Is it the endocrinologist? The neurologist who happens to be seeing them? The ophthalmologist who doesn't realize that you've already covered those genes of interest, but they've got their panel? And you have to do an informed consent discussion before you order the test. You also have to have a plan in place for how are you going to send the results back to the family, because these are complex results. It's not a 5-minute job to do this. Then the question becomes, what test to get? Bigger is not necessarily a better test, and you still have to get it paid for. You can sometimes get a bigger test paid for if you have a good reason for needing the bigger test, case by case basis. So how has molecular analysis changed diagnostics? It's changed medicine in general. We can confirm clinical suspicions. I think you've got hypophosphatasia. Well, that's a clinical diagnosis. But you can't predict for younger members of the family who don't have early dental loss until you test and test cascade-wise family members. You can dissect out genetic heterogeneity. Everything that quacks is not necessarily a duck. Some of them are geese, some of them are swans with laryngitis. So there's a potential to improve your health outcome. But the challenges with doing molecular testing: what's the reliability of your test? How accurate is the test interpretation? And that's at the technical aspect within the lab, and in the interpretive aspect, which is you've got a true result, but can you actually get it all the way to your patient? There's a potential for harm when you do genetic testing. You can have variant misclassification. You classify sequence variant as potentially pathogenic. That is physical and psychological harm to the patient. Physical because you're going to do things to monitor a disease. So, agents as simple as blood draw, that's still harm over time, one of more invasive things. Psychological, you've got the vulnerable child, vulnerable family syndrome to worry about, and they never let go of that diagnosis. You can have uncertain results, and if they're not prepared for those uncertain results, they get turned off to current allopathic medicine. You may find incidental findings. It's not what you were hunting for, but it's something that's significant. How do you deal with those? How do you get those back to the family? How do you get them where they need to be to address those other concerns? And all of this is part of the psychosocial and social impacts of doing genetic testing. So when you decide to do a genetic test, you need some considerations of the benefit for the patient. One good therapeutic options keep being inferred. Yeah, they've got this disease, it changes my management. That's what the insurance company wants to hear. Because that's just about guarantee you all right, we'll let you get it this time. But not all tests are feasible because it may require invasive techniques to get the tissue of interest. If something's not expressed of one tissue or another, or with COVID, you can't get your patient to a blood draw station. The station's closed down, the family's not going anywhere. Oh, and the lab won't do a buckle for that one. Test availability. I've got one I'm going round and round on. The test is available, but not in network for their insurance. And it's only available in a panel. It's not available as a single gene anywhere in North America. Yeah. What's the informational value? How much does it change your management today, tomorrow, into the future? Think of that one before you order. And what's the cost effectiveness? You can order a panel, but if your yield for your patient is very tiny for a very expensive panel, is it worth doing it? What's the cost to the patient? What's the cost to society for doing a genetic test? And in general, and certainly for those who've been trained in Europe, predictive genetic testing without therapeutic consequences is not indicated. So geneticists take that to heart. If it's a disorder that is not going to show up until you are of legal age, we will not test for it while you're under 18. The day you turn 18, you can walk into my clinic and say, remember me? Oh yeah, you're so-and-so's older brother. I want that genetic test, okay? Boom. Do the counseling, I'll order it right then, and I'll stick the family with the bill because they've heard this before. Not until they're 18 and it's free will to get that testing done. It's Pandora's box. You can never put that information back into the box. And you have a right to stick your head in the sand, to not know. And that's an ethical right that geneticists try not to violate. Okay. So what's appropriate testing? Not everything has to be molecular. Karyotypes still have the utility, certainly for you guys. Mosaicism analysis, which you have to order as an extra, has utility. There are a few times where a fish is good enough. A fluorescent in situ hybridization, if you're looking for one gene, one change, or an area of copy number variation, where there's 3 copies of something, it's a duplication of a little area, a fish can tell you that. You can do an array. Comparative genome hybridization, aka chromosomic array, which happens to be a formerly trademarked term. You can sequence single, multiple genes. You can do genomic sequencing if you want to pay for it. No insurance is currently covering that on a routine basis. And there's even chances on a research basis, slowly being translated into clinical basis, for being able to do RNA sequencing, which actually tells you more some of your variants. So what are the prerequisites for doing targeted and efficient genetic testing? You gotta have recorded history. The medical history's gotta be recorded. It's gotta be recorded adequately. You need a karyotype, and the standard of care is to use the Human Phenotype Ontology, hpo.jax.org. It's free to get it, okay? Because this is what the folks in the lab who are not staring at your patient, who are not seeing the pictures of your patient, are going to use to go, yes, we're going to filter that gene in or filter that gene out. Okay. Funny looking kid does not count. You have to say what and why. If it exceeds your capacity within the time frame of clinic, get some help describing the child. Send them to your geneticist. In the ontology, each term describes a specific phenotypic abnormality. So it's one-to-one mapping, which makes it real easy for doing artificial intelligence-based filtering. Do a pedigree, at least 3 generations, ideally 4 generations. Record the pedigree. If the pedigree is not recorded, it's not any good to anybody. Ideally, you do it in standard notation. I'll take somebody's longhand notes that go, one older brother, healthy, younger sister, ADHD, mom's got this, grandma's got that, and on the other side, we've got grandpa this, 3 uncles with this, 3 aunts with this, and all the first cousins are healthy. Okay, that gets me a start. It'll only take me 40 minutes to get the rest instead of an hour. Okay. And as you folks know, let's really preach to the choir. Genetic testing is not necessarily the first step. You've got specialized endocrine approaches. You've got baseline testing. You've got dynamic testing. You can do functional imaging and specific hormonal analysis, tools that aren't necessarily available on the other sides of the street. You also have to do informed consent for DNA testing. Signing the form isn't good enough. You have to explain to the family what they're getting into. Okay. So a simple algorithm, and I've adapted this one from Eggerman, is you got a clinical diagnosis, you get your multidisciplinary evaluation. You can do it all yourself, great. Get the family history, look at the imaging, do that pre-test counseling, information to the family about what the test is, what can be found, and document your informed consent. You need a signature. If you have a specific phenotype and it's only associated with one locus, one particular gene, you can do a disease-specific test. It's a one-to-one mapping. If it's heterogeneous, cytoma, etc., okay, then you do a multi-gene panel, because otherwise you're going one bite at the apple, you bring them back in 2 months, do it again and again. That's great for the RVUs, bad for the patient. Okay. If you don't find anything on a multi-gene panel, it's time to broaden your search if you still think it's genetic. And that's where you go to a clinical exome. No, it's not a whole exome because we don't every exome. Quote, whole genome sequencing, or something called long reads, which will get things that are not in the code sequence in some cases. And that's time to go back and forth with the folks in the lab, bioinformatics, and maybe time for round 2 of a discussion with somebody who plays on the molecular side. So what are you going to get back? You're either going to get back pathogenic variants, no variants, a variant of uncertain significance, or you're going to get something from incidental findings and secondary findings. And that's why you have to do the post-test, the counseling, potential interventions, and the potential for cascade testing of other family members. So we'd like to have some guidance. The ideal guidance would be international consortia with evidence-based recommendations. Not many of them. Fair kinds of guidelines would be consortia, primarily evidence-based and a little bit of, yeah, this is the way we've always done it. Not ideal, evidence-based. The grayer your hair, the more weight your word has. Curbside consults, not good, because that's an off the cuff, and they may not think of all the things that need to be thought of, and they're certainly not going to help you with counseling. Okay? There's a lack of guidance within the US. I'm going to pick on the Endocrine Society. I am a card-carrying member. I can do it. There's a pediatric obesity guideline. It is short on details for genetic evaluation. That is probably because science is going faster. However, the primary adrenal insufficiency guideline doesn't mention genetic testing at all. The ACMG doesn't have anything. So geneticists haven't come up with guidelines for testing for endocrine diseases. We've got some guidelines for some other things. And the Pediatric Endocrine Society co-sponsors the obesity guideline, but hasn't put anything out that I'm aware of on their own. But there are guidelines. The Europeans are ahead of us in doing some of this. So here's a perfectly good one for molecular diagnosis and the management of differences or disorders of sex development. Written by experts, so they are clinicians, they're laboratorians, and it's a consensus guideline, and it lays out steps 1, 2, 3, 4. Karyotype, copy number variants, steroid analysis. Then you go to a multidisciplinary team to do that deep phenotype, the rest of the biochemical, other genetic evaluations, and they use a high throughput technique. High throughput means next generation sequencing, it means a panel, it means exomic sequencing. Okay? And they reserve genomic sequencing if they think about novel genes and things that are polygenic, things that they haven't been able to solve going through the rest of this pipeline. So if you've done a test and you feel confident you ordered the right test, now you have to get results back. So the first thing, I run into this all the time, seeing results from outside, have they given informed consent? Does the family understand the limitations of the testing that you're doing? Do they understand the categories of results? Pathogenic, likely pathogenic, uncertain, benign not necessarily equal to, except in their mind, normal. Who's going to explain the results? Did you order it because it's a 6-month wait to get into your local geneticist? And have you talked to the geneticist before you ordered it? And the geneticist is going to accept the responsibility for returning those results? Or are you going to return? In what setting are you going to do this? My clinic, it is face-to-face for doing return of genetic results. It is never telephonic. It is never, we will mail these to you. And I don't care what the education level is of the family receiving the results. That is just our guideline, face-to-face. In selected cases, currently, we are doing it as a secure audio video, telehealth. Because you're going to have to answer the questions in real time. Okay? So what are your possibilities? It's true, it's pathogenic or likely pathogenic, and it's relevant to why you did the testing. It's pathogenic, but it's not the reason the testing was done. Benign, which doesn't mean no disease, means no disease found with whatever test you did. The dreaded variant of uncertain significance. Or you found a single variant in a disorder whose inheritance pattern is autosomal recessive. That's one hit, you need two. And you have a willingness to reassess the results over time. Results and interpretations do change. So of course, we know the answer. We slightly found the reason, thanks to Jerome Horowitz. If you found what you were hoping for, you got clearly pathogenic variants in the gene of interest, and it's consistent with the phenotype, then you hope it's proven biallelic inheritance, because you could have two pathogenic changes on one copy of the gene, none on the other, and for a recessive disorder, that does not give you full phenotype. You're looking for 1 and 1, which means you have to have a mechanism for being able to do the parents to prove that you have them in a trans configuration, one on each copy, rather than in a cis configuration. What you hope you don't see is multiple pathogenics in the same gene, 3 or changes, or single changes in multiple candidate genes, which is the problem with doing parents. You get a hit there, a hit there, a hit there, then what's really causing disease? And we all have a lot of variation. Estimation that we're all carriers for at least 2 pathogenic alleles, and 90% of the pathogenic alleles were in the top 100 genes. Looking at the Dutch and Estonians, probably similar numbers for folks in the United States, but a lot of those other variants were VUSs, and at the estimate of 20%, all the other variants being VUS is conservative. It's probably higher than that. So you can get pathogenic, yeah, that's disease causing. Likely pathogenic, just as it sounds. Probably causing disease. Uncertain, likely benign, or benign. And these come as the terms from the ACMG guidelines, the 2015 guidelines, which are conservative guidelines. It's a weighted matrix to have the lab use all the information available to be able to give you a 1-word or 2-word response to, does this mean anything? It gives you a reduced potential for false positives, and it results in way more variants of unclear significance. But it's the best standard we've got. It uses variant classification, population frequency data, family segregation data when available, and heavily is weighted towards functional data, which were in short supply. So erring on the side of caution, magnifying our VUS problem. Every time you add more genes, you increase your VUS problem. Okay. So clinical genetic testing is usually taken in individuals who have a high a priori probability of having a genetically mediated disorder, as opposed to, oh, we're going to go screen the next 500 people on I-35 out here for variants in X, Y, or Z gene, and what do those variants mean? Unless they're on their way to one of the healthcare centers, those variants probably don't mean much of anything. Okay, so what's a variant of unclear significance? Sometimes it seems as simple as interpreting the volume under a surface, which is everybody recognizes as a double integral. I don't want to have to do that. And sometimes it seems to have the impact of the vehicle uplink system, which is where I got the idea for the title of the talk, because if one of these things hits you, you've been run over by a VUS. So what about the clinical use of variants of unclear significance? The ACMG guidelines say never, ever, ever use them for clinical decision making. They're identified. At least 20% of all genetic tests. And it doesn't matter whether you do a single gene to a multi-gene panel. The more genes, the more VUS. So there's actually data. Almost 12,000 variants in a commercial reference laboratory, still in existence. And they scored over 50% the variants that they found as VUSs. If it was a private variant limited to the family, they didn't find anybody else, it was 80% of the time it was called a variant of unclear significance, because we just don't have enough information to reclassify it. VUS results are returned to families, and depending on what they've heard and retained by the time they leave your office and get home, they may take that as, oh, that's disease, or no, that variant doesn't cause disease. The answer is somewhere in between. So VUSs are never used for clinical decision making, right? I violated that one. Okay. So resolving VUSs. How well does your phenotype fit the genetic information that comes back? Does it fit? What is the VUS? Is it in a section of the gene, or is it a gene where it's a loss of function or a gain of function? So you may have to know something about the gene, the protein, and the protein's interactions, the domains of the protein, in order to be able to interpret a variant of unclear significance. It gets to be more fun when you have two VUSs in the same gene. So what's the inheritance pattern? And are the variants, again, in cis or in trans? Makes a big difference. You can have them in cis and each one by themselves, not causing much of a problem, but the pair in combination becoming pathogenic. Okay. So what lines of evidence are used to help resolve this? The more independent lines of evaluation that you have, the better off you are. First one is biomarkers. Okay. You have an abnormal laboratory test. If you think it's a gene for short stature, you have a short patient in a ruler. Okay. Something like that counts as a biomarker. Multiple family members. Ideally, multiple family members across generations, if you've got dominant disease or X-linked disease, or a big enough SIB that you've got multiple family members in the same generation to go, aha, every family member who has disease has this sequence change. All those without that sequence change have no evidence of the phenotype. That gives us some evidence that it's corresponding to disease. Or functional assays. Well, in some cases, you can do enzyme assays. And you might then convince somebody to get an invasive biopsy for a tissue where you can actually analyze a particular gene. Is there an animal model? If we've got a naturally occurring animal model that has the same phenotype, then that gene's defect at the same position is probably causative. Can we build a knockout or a conditional knock-in or any of the other variants that we can do in animal models? Well, great academic centers, not so good for the large majority of individuals, because you need a big facility behind you in order to do that. You can also look for conservation across species. If you have an invariant arginine from yeast through Drosophila, through all the chordates, through all the primates, Mother Nature probably needs that arginine there for that protein to work. So a change to anything else probably means something. The wider the range of species with that conservation, the more evidence. And of course, the more lines of these evidences, which are almost independent, the greater the probability that VUS has pathogenic significance. So are there tools to resolve this? Well, the lab used one tool before they return them to you. They use the ACMG guidelines. None of the reference labs will do less than that. They'll use various databases. Easy to get to. ClinVar. Your taxes paid for it. It's out there. You can get it with 4 clicks in Google. HGMD, Genomics, which is great for looking at populations, because this is referenced on individuals who were normal. So how frequently does this particular variant show up in a normal population? They're individuals who don't have disease by definition, and they do have some ethnicity distribution within that, which may help if you've got a variant that only shows up in individuals who claim Hispanic, Latino, Latinx ancestry and you got it. There are also predictive in silico algorithms, and the different algorithms weigh different factors differently, and two algorithms may not agree. Back when most places were only using two algorithms, you get, and the algorithms don't agree. So polyfin, whatever they were using. Those algorithms don't help in splicing. You have to use the splicing algorithm, and there are several of those out there as well. Splicing is that invariant 6 bases on either side of where you snip out an intron and glue those 2 exons together. A change in those is generally going to cause a change in coding sequence. More so if it's position 2 than at position 6. But sometimes you have a deep change and it causes a new splice site, and a splicing algorithm will pick that up and say, oh, possible new splice site. Okay. Some of the algorithms do incorporate some of this phylogenetic, i.e. it's invariant across multiple different species, or physical chemical difference, because they can read the chemistry books. But their accuracy, 65 to 80%. Algorithms are better than coin flip, but depends. Okay, so how do you resolve the rest of them? Consortium. In some cases, the reference laboratory who sends you the results says, this is an interesting VUS, and you know what? So-and-so in Europe is interested in this gene. Talk to them. They may have some tools to be able to sort this out. There's a huge consortium in the United States. We're doing it. Undiagnosed Disease Network. Federally funded. Closest center to us is Baylor, Houston. Okay. You can bundle up your patient, all their records, and they will elect to see your patient. They do not take everybody. It's a research-based. If they find something, they'll let you know. And they've got more tools than most everybody else because they use tools all across the US to look at one patient. Okay. The cloud has some advantages, and social media has some advantages because you get cloud source variant databases. This family goes online on Facebook and says, my kid's got these features and they think it's got this, and you suddenly get something fired back from Sweden. Oh, my child's got the same thing. Boom, boom, boom. And pretty soon you go, it's not a variant of unclear significance. It's a subpopulation we didn't realize that is worldwide and spread, who truly have a disease, and they all have the same sequence variant. And there's not much phenotypic variation. Now you know that's probably pathogenic because it wasn't just our lab that did it. There's a bunch of labs that came up with the same interpretation, and the families beat us to the best interpretation. Okay. We'll try getting functional data. That's easier if your sequence variants in the catalytic domain, i.e. an enzyme works, doesn't work, works too well, than it is in a signaling pathway. Endocrinology, yeah, he's got too many signaling pathways. Cell systems. You put the variant into a cell system, expressed normally versus overexpressed, makes a difference. See what happens. Does the cell phenotype? Something not localized right, tracked right, so that you can do that. Or back to the animal model systems. There are some commercial tools. Genomics.com allows you to double check the commercial lab's results because it runs your variant through publicly accessible databases. It does part of the lab and library search for you because it pulls up if your particular sequence variant is in a publication, the beauty of AI, and goes, there's one paper, boom, and it links you to the PubMed reference. So you can at least read the abstract right there, okay? It's not the only tool out there. If you like playing on the federal sites, you can use Proven, and you can get a lot of the same things out of that. Then you've got to put the clinical input. How stringent do you need to be? Okay. Are there other variants, the same nucleotide, the same triplet, do they cause disease? If it's not the exact same nucleotide and the exact same change, ACMG downgrades it. But I had a case with a change of a methionine to one of the branch chain amino acids. CLAR had the other two branch chain amino acids as being pathogenic. They didn't call mine. I looked at my patient and went, yeah, it fits. It's a good fit. I called it. And I said, based on my interpretation, if these others are pathogenic and they're clearly pathogenic, they've got enough support, then this one's pathogenic. It's just not been gone through the same rigorous kinds of things. Okay, so that's great because you had one pathogenic variant, one non-pathogenic variant in your autosomal recessive disease. You got it solved, but what if you only have one? One of the possibilities is that you've got an intronic variant. Why? We only sequence 10 to 20 bases into that non-coding region. Okay? You can have a promoter variant. Good example in galactosemia. The Duarte variant of galactosemia is a promoter variant, not the polymorphism at position N314. You can have a 3' variant, or you can have a deletion. Okay? All those exist. This is not one and done medicine. Reclassification, upgrades, downgrades. Most of the time it's a downgrade. Only 9% of the time, so it's an upgrade to disease, but there's better filtering, and this goes back to reinterpretation of the data. In some cases, you'll find something new, like splice site variants. There's a paper that I'm on that just got accepted, novel splice site variant in a novel disease. Okay, so this is really the summation. Okay, we've been analyzing the results of your genetic screen, and I'm happy to say there's no cause for any concern, as we discussed, although this isn't an exact science. So you balance the expectations. Okay, this is a good paper. Why? Because it picks on one of the diseases I picked on before. It's an endocrine paper. Summation: 1 in 3 times when they did what was state-of-the-art genetics the day they saw the patients, you didn't make a genetic diagnosis. So the families expect yes, no. Sometimes you get a maybe. Reanalysis and reclassification can take months, years, or decades. So do your homework. So there's your summary. Do the right test at the right time with informed consent. Review the results. Have your ducks in a row before you talk to the family, and have a plan for going further. References, they're all available. Thank you very much for your attention. If you do a panel and you get multiple VUSs on your panel, what do you do with that? You look for the subtle differences in the phenotypes between the various things on the panel. That's the first step. You look to see, do you have 2 variants in the same gene? What's the inheritance? If you've got one change for an autosomal dominant and one change in an autosomal recessive, how frequently does the dominant one have variants versus the recessive one? You had to do your digging for your homework there, and you might go, they got disease, it's probably this, because we can't find any of the other variants to be the second variant for those recessive ones. But you go back and you look at it again. Next time they come to clinic, a year later, and after a couple of years, you do get the lab to relook at this, whether it's no cost or some cost, because the data there, they'll reclassify it. There are some readily available ones. Some of the reference laboratories have consent forms that are available. The one that we use when there's no consent form is essentially one we've made up that says you may get results that don't give you a clear answer, and you understand that we're not going to return these except in a face-to-face office visit, period. And if they sign that, you've got them where they can't argue because they signed it as informed consent, that you may get an answer, you may not get an answer. There may be technical problems, such as the lab says inadequate specimen, something happened to the specimen along the way, there's a hiccup. Yeah, it's all in our homegrown one. But we've had that one for over a decade because some of the labs didn't have anything.